Good afternoon, everyone, and welcome to the Cancer Research Foundation's second virtual event this fall. My name is Matt Johnson, and I am CEO of the Cancer Research Foundation. Thank you all for joining us this evening. We are extremely excited to have Dr. Charles Kaufman and Dr. Amber Stratman presenting tonight from Washington University in St. Louis. Thank you, Chuck and Amber, for being with us.
Thank you.
For those of you that may not know, the Cancer Research Foundation's mission is to support early career researchers and novel ideas in cancer research with the hope of contributing to transformational events in the prevention, treatment, and cures of cancer. We invest in high-risk, high-reward ideas early on that may be difficult to fund through traditional sources. So with that, a reminder to please type in your questions in the Q&A on the bottom toolbar, or once we get to the Q&A portion, you can raise your hand. There's a raise your hand button, and you can ask your question yourself. And I'd now like to introduce Zana Nikitas, who is the Cancer Research Foundation's board chair, to introduce our presenters. Zana?
Thanks, Matt. To keep things moving along, I'm going to introduce both our speakers first, and then let each of them present to all of you directly. We'll have time for questions at the end. Our first speaker will be Dr. Charles Kaufman. Dr. Kaufman is an assistant professor in the Department of Medicine at the Washington University School of Medicine in St. Louis. As an oncologist, Dr. Kaufman sees patients with melanoma and skin cancer, and in the lab, Dr. Kaufman pursues research in cancer initiation and development, epigenetics, and developmental biology. Dr. Kaufman was awarded a 2016 Cancer Research Foundation Young Investigator Award to investigate melanoma initiation. Welcome, Chuck.
Thank you so much.
Second to present will be Dr. Amber Stratman, an assistant professor in the Department of Cell Biology and Physiology at the Washington University School of Medicine in St. Louis. Dr. Stratman studies the mechanisms that regulate the development of blood vessels and how a body stabilizes and regulates blood flow in both development and disease. Dr. Stratman was awarded a 2020 Cancer Research Foundation Young Investigator Award to explore new ways to stop tumor growth. Welcome, Amber, and thank you both for being here. With that, I'm going to turn the spotlight over to Dr. Kaufman.
All right, thank you so much. And I'll bring up my presentation here. Okay. All right, looks like everybody can see things. Great, thank you. Well, thank you so much for having us tonight. And I very much appreciate the support of Cancer Research Foundation. And I'll talk about the work that's been supported and how that's led to a number of our other projects. So we're interested, in simple terms, how and why does cancer start, and how and why specifically does melanoma start? So melanoma, just as a little bit of background, is a skin cancer. It arises from melanocytes, which is the term for the pigment cells of the skin. And melanoma is unfortunately a pretty significant cause of cancer death, with about 10,000 folks dying each year in the US from melanoma. It's one of the few cancers whose rate is rising, thought in part to be from things like tanning bed use and intermittent sun exposure. And historically, the real alarming features of melanoma is that it's tends to spread or metastasize. In the past, the few treatment options weren't that great. We now have some really great treatments, but for some folks, they don't work and they can have some tough side effects. And so really what we approach things is this sort of long game idea that some basic, sort of fundamental research leads to new treatments. And in melanoma, that can be seen where for quite a long time, back to the mid-70s, a chemotherapy called dacarbazine was really the only treatment. And there wasn't a whole lot, a hint of immunotherapy in the late 90s. And then within the last 2 years, an absolute explosion of treatments, many of these derived from work from an immunotherapy for which a Nobel Prize was awarded recently. And so we do hope we can do better for patients, and we want to do better. So our work was spurred on by this idea of how does melanoma start? And if we could see that first cell of melanoma, could we come up with some new approaches? And so briefly, a melanocyte is this pigment cell that can become a mole, a small spot on the skin. And most of the time, moles are fine. They don't cause any trouble, but very rarely, way less than 0.03% in one's lifetime is any one mole at risk. But sometimes a cell within that mole can become a cancer cell and go on to form melanoma. And so using the zebrafish, which I'll tell you a little bit about, we put one of the major mutations in human melanoma called BRAF in that fish using transgenic techniques. And those fish go on to form tumors over the course of months. Every fish will get a tumor, but it takes many months. And so that allows us to try to understand the early process. So why zebrafish? Well, zebrafish are small. They're about this big. They're vertebrates. So like us, they have a spine and they share many of the same tissues. So everything but lungs pretty much are there, and blood, and importantly, pigment cells are melanocytes. And when looking at the genes involved, they share greater than 70% of human genes that have a zebrafish version. So briefly, I just wanted to take a little side trip in how disease models can help people using fish. And this is from a story that came out last summer. And I want to highlight, this is not our work, but was just such an amazing example of how a fish or an animal model could be used. And this were folks with a mutation in this gene called ARAF. And so what happened in this situation where these children who have fluid retention, where from the paper, this child has swelling in the legs, and this is a problem caused by lymphatic overgrowth. And so this group found the gene that is mutated that causes it and put it in the zebrafish. The fish got the same disease. These fish had excess retention of fluid. And then they were able to test many candidate drugs very quickly and found that not only do the fish grow fine, which was important because if you're going to give a drug to a child that's growing, the fish grew okay, but it fixed the disease in the fish. And that led the FDA to agree to let them try this drug in this kiddo and fixed the lymphatic problem. And you can see pretty graphically here. And so really an amazing kind of home run scenario of where you're going from basic science to a treatment using the fish model from there. So back to how would we ultimately like to apply such things in melanoma? So we asked this question, if only we could see melanoma, the first cell. And so in my postdoc work, I developed a fish here, which I hope you can see the video swimming around with a melanoma here on its back, and created a beacon or a fluorescent reporter of melanoma here on the back of that fish. So that allows us to see that bump. Now you could obviously spot the bump even without the fluorescence, but the real trick here now is we can now see these tumors when they're very small, even at the first cell, and contract those over time, and allows us to understand better how that is controlled. So how are we going to use this to improve cancer or treatment in people? So if we can understand how that beacon is controlled, we can understand how melanoma starts. Maybe we can prevent it from coming on, and perhaps we can find other changes that occur at the same time and allow for earlier detection, and then also perhaps block the underlying causes and provide new treatments for melanoma, maybe new drugs, new interventions, which I'll mention at the end, things like diet. So briefly, the work that was in part supported by Cancer Research Foundation, we wanted to understand what happens in this transition from a melanocyte to a melanoma. One of my graduate students, Eva Kramer, developed this fish that has red melanocytes and green tumors. And so this is what that fish looks like in red. So the whole fish is red, and you can see here in the zoom, here's a melanocyte. There's pigment here and red around it. And then that fish has a tumor here and a small early tumor here in green. And so she was able to sort out those cells and look at the genes that come on and the chromatin shape changes that occur. And what that showed us with a number of techniques, one of which being called a TAC-seq, was that there are consistent changes in the open structure of chromatin, near very important genes in melanoma like SOX10. what this represents is these little peaks are areas of the gene that are open, that are accessible for regulation. And that suggested that there's a sort of a stereotyped and tractable epigenetic path to cancer, something that we could study and hopefully interrupt. So Rebecca Cunningham, another student in the lab, then went on to use this information to study each of these areas, these little peaks of open DNA, to see how they are regulated in the developing embryo and in tumors. And kind of cut to the chase, what she found was that a very small region near this gene SOX10, just a few hundred little ATs, Gs, and Cs, allowed us to mark these tumors in the same way. So we found another tumor beacon, and it's providing a more direct link between the zebrafish and humans, since this gene SOX10 is highly conserved between fish and humans. So from here, this has suggested a model where, from other work as well, that SOX10 seems to be turning itself on in the transition from a melanocyte to melanoma. So this gene SOX10 here, it makes the protein and it goes back and auto-regulates. So it's like a feed-forward loop. So we'd like to understand how to interrupt that process and potentially treat melanoma. So going forward, we're using things like CRISPR, which again was a Nobel awarded a technology this year to delete these regulatory regions in the fish, and then also in human models, cell line models. And then one thing we're really excited about that Katie's working on is finding partners of SOX10, so other genes it interacts with, that may provide targets for drugs that could interrupt this process. One of the other really exciting things that came out of this work, and sort of the serendipity of science, when we were doing this project, and I should mention that the work I just mentioned is in a preprint that we have out and is in review right now, which we're excited about. So we noticed that in our facility, our zebrafish facility, we have these robots, this gizmo here that goes around and feeds the fish for us. We noticed that our fish get fed a lot more than in where I trained, and the fish were getting tumors faster. And so we looked at that systematically and realized that as you increase feeding, you increase the speed of tumor formation. And so that shows up here, in this blue curve is showing how many fish have tumors at a given time. So the blue line here, half the fish have tumors at 180 days when you feed them 4 times a day. When you feed a fish once a day, they still don't, even at 260 days, not even half of them have tumors yet, which we found to be really exciting. And beyond that, we found that because we can see that first cell of the cancer, not just a big tumor, that we could even interrupt the formation of that first cell of cancer. So when you feed these fish 4 times a day, twice a day, they get the first cell of cancer. But when we fed them once a day, it really delayed the onset of cancer, even at the single cell level. Interestingly, even intermittent fasting, I know that's sort of a hot topic these days for folks, and we can intermittently fast these guys, and it also slows the onset of the cancer, which we also, is sort of an unexplained but well-known phenomenon in other scenarios. And so that spurred on some other folks in the lab then to look at, is intermittent fasting, things like that, not only protective, or are they actually protective against persistence of these early melanoma patches? And so John Spalding, a grad student in the lab, with an undergrad Alex and another technician Brennan, have been working on this. And just a brief snippet. So we found that here are these, kind of beautiful but also ominous early cancer patches here on the fish. And if you fast that fish, which 2 weeks is a long time for a human, but for a fish is nothing. It'd be like a day for us, the way that they eat. And that tumor patch largely goes away. And in fact, we have examples where we can fast the fish, and that tumor patch 2 weeks later has gone away, and you feed them again and it stays away. So it's really completely, we use the word cure carefully, but it's gone as far as we can tell, which is really remarkable. So in an unexpected way, this has really, opened up some other avenues of research where we can now try to understand how, what happens to these cells. there are, processes by which cells can go away, via apoptosis. Are they using a process called autophagy to regress? And then what happens when we refeed them? This one that was essentially cured was super exciting, but on occasion we'll actually see some that almost go away, but a few cells remain, and then they grow fast after we feed them again. And so understanding what's special about those bad players would be really informative. And then we're really getting into the ability to treat fish with drugs, as I showed you, and then manipulate the genetics of the animal in a fast way, what exactly the endocrine pathways are, metabolic pathways involved. And so, ultimately other drugs that could achieve a similar effect. So with that, and I know we'll do questions later, wanted to thank the folks in the lab. I think I've highlighted the folks along the way. Current lab members, I've highlighted some former lab members. This is a logo that one of my grad students put together, who's an incredibly talented artist. And we say we put the BRAF in zebrafish, because that's that gene that causes the melanomas. And here again, thanking the Cancer Research Foundation, and more, that support has really led to other successes for us in getting funding from the Melanoma Research Alliance, NCI, and then other organizations. And finally, again, just this is our group in happier times when we could meet in person and decorate zebrafish cookies at my house, visit the aquarium in Boston where we wear our matching t-shirts. But now we're largely on Zoom meetings these days with our furry animals and such. So thank you so much, and I look forward to questions after Amber speaks here. Thank you. Give back control of the screen sharing.
Okay, before I pass it to Amber, I just want to remind everybody, Q&A, you can type in your questions now if you have some for Chuck, or just remember to send a few questions. All right, Amber, to you.
Thank you. Get this going here. I want to reiterate what Chuck said and just say again how happy I am to be here today to present our work to you. We are a fairly new lab. We just opened at Washington University in St. Louis in the School of Medicine at the end of 2018. And so we're really just getting going on our work. And so it's been invaluable to have the support of the Cancer Research Foundation to help us explore high impact, what we hope are high impact and high risk projects while we're really starting to form the direction of our lab. So today I want to tell you about our work that we do in studying the tumor microenvironment. And so for us, this includes blood vessels. And let me get this going here. And so why study blood vessels in the context of a tumor? So in general, the reason why we're motivated by this concept is that tumors need a blood supply to exchange oxygen and nutrients, just like any other tissue in our body. And the idea was put forward a number of years ago, and we'll touch base on this in a little bit, that if you could starve a tumor of its blood supply, you would be able to starve it of its ability to grow. And this concept was really put forward by one of the seminal people in this field, Dr. Judah Folkman, in the 1970s. And he's really considered the father of this concept of tumor angiogenesis. And what he proposed in his work was a few things. One, he recognized that tumors didn't grow past a certain size without a blood supply. And he also intimated that tumors were growing new vessels to get that blood supply. They weren't just co-opting vessels within the human or the animal. And then he, from that reason that the tumors must be producing a factor that caused blood vessels to grow into the tumors. And then ultimately, they proposed the concept that if you block the blood vessel supply growing into these newly forming tumors, you should be able to also stop tumors from growing and essentially starve them out and regress them, stop them from spreading around the body and metastasizing. And so this is a great concept. And so again, just highlighting what that concept is that, in this case, the molecule that makes blood vessels grow is VEGF, the primary molecule. So the concept is if you treated the tumor microenvironment with an inhibitor to block the blood vessels, essentially targeting VEGF, perhaps you can make these vessels smaller, shrink the tumor, and prevent metastasis. And there have been many clinical trials surrounding this point, and it does work for a little bit. So you can treat tumors, these are approved in people, you can treat them with VEGF inhibitors. They do regress the vessels out of the tumors and the tumors start to shrink. But what happens is over time, the tumors essentially say, I don't care. I don't care that you're giving us this anti-angiogenic therapy. We'll just make more VEGF. We'll make more of the other things that cause blood vessels to grow. And what happens is these tumors, they come back larger and more aggressive than they were before. And people typically die from metastatic cancer. And so the field is still really searching for a targeted way to address this problem. And so our question is really motivated by, is there anything we can do about this issue? And so we use the zebrafish, partially for many of the reasons Chuck highlighted, because they're small, they're high throughput, their biology is shockingly similar to humans. But the other thing I want to highlight here is that we have exquisite ability to label individual cell types in the fish. And you can see that here in these different images. So here's all the blood vessels labeled in this fish in the center. We can label blood, which you're seeing in these panels to the left in magenta and the blood vessels in green. And then we can do genetic manipulations on these fish and see if they affect the ability of blood vessels to grow too much, as you're seeing in the panels in the bottom right, or too little, like you're seeing in the panels in the upper left, which is what we're going to be talking about today. And to just highlight how powerful this is, you can actually watch all of this happen in real time. So this is how a zebrafish is born. They're born as these single cell embryos. And then over a period of, let's say 22 hours, you can put the fish under just a very simple dissecting microscope. So any lab could buy this equipment to do this, which is the other beauty of this. And you can watch the fish actually develop from a single egg, a single cell, all the way up into an animal, a multicellular organism. So here's the head and the eye forming down here. This is the muscle in the back. This is the tail of the fish. And this green is new blood vessels forming. So we use this as kind of a small incubated bioconductor, if you will, for what a tumor microenvironment might be like. So if a fish can grow a new vessel, perhaps the tumor does it in the same way. And so that's how we started our studies. So again, just to highlight, this is what our transgenic fish looks like. This is all the blood vessels in the fish. This fish is 5 days old. And let's see, right here in the trunk, kind of this midsection of the trunk, is where we focus most of our studies. So we did a genetic screen. We randomly mutagenized the genome and we looked for things that blocked the fish's ability to grow new blood vessels. And when we did that, we pulled out this fish here that you can see. This is the wild type sibling. They have these, green blood vessels that go all the way from the bottom of the fish to the top. And that's really what we're looking for to call them normal. In the mutant condition, you can see these vessels don't do that. They go a little bit and then they stop. And so we were very interested in this candidate because it seemed to be specifically targeting these vessels that were forming. And so this turns out to be a mutation in a gene called CDS2, which is not important for anything we're going to talk about today, other than to say it is required to resynthesize this downstream molecule called PIP2. And so when, for the rest of the talk, you can just know that we're going to call anything that inhibits this circular pathway a PIP2 inhibitor, and I'm happy to get into more details in the question and answer if anybody has specific questions. So the first thing you should know to understand the concept of the story and what we're really driving for is that pro-vessel signals, so we'll call them pro-angiogenic signals, like VEGF, drive vessels to expand. So this is what the control looks like, and this is a fish that has more VEGF in it, so like a tumor environment. And so when we do that, the vessels start to grow, they start to expand, they go into new areas that they typically aren't in. So again, this is what our mutant looks like, zoomed in on that mid-trunk area. So these are the vessels that go from the bottom of the fish here to the top. This is what the mutant, they stop about the middle of the fish. And so we reasoned, if VEGF makes vessels grow more, perhaps it would make these vessels grow more in the mutant background. But what actually happened is when we put VEGF in this mutant background, the vessels got worse. So not only did it not make them better, it made them aggressively worse. And so now we had a fish that had no blood vessels in it. And so why would this matter to us? And this really goes back to why treatment with VEGF inhibitors fail as a tumor therapeutic. So again, you know, the tumor makes VEGF to call in new blood vessels. You can treat with VEGF inhibitors, it gets rid of the blood vessels, but then the vessels come back more aggressively. And because there's much more VEGF made by the tumor. But now we had an instance where we had a condition where more VEGF was making blood vessels worse. So we reasoned if we could target this pathway, we could use the tumor's ability to make more VEGF to try to work against itself. So again, this is what this looks like in the fish. And all of this was kind of conceived and conceptualized starting with the zebrafish. But to really get it into a tumor model, we decided to take this to the mouse. And so what we did is we made a genetic knockout in the mouse for the same gene that's missing in our zebrafish. And then we implanted allograft tumors. So we allografted in either lung carcinomas or melanomas. And what you can see, the yellow line here is our mice that are deficient for this gene of interest, CDS2. And all these other lines are various controls that we did for these studies. But I think what's most impactful is if you look at these tumor images over here on the right-hand side, you can see by just taking out this single gene out of endothelial cells, we can reduce the growth rate of these tumors. And what's really exciting to us is when we look inside these tumors, if we take a tumor out of the mouse and cross-section it and look inside it, what you're seeing over here in this green staining is that the blood vessels are almost completely gone. And that's quantified in this graph here. So this yellow box is showing that the blood vessels are greater than 50 or 60% depleted out of these tumors. So this was very exciting for us. But of course, we're not going to make a genetic deletion in humans to try to recapitulate this finding. And so we worked really hard to find a small molecule inhibitor. We actually found one that was in clinical trials for schizophrenia, because schizophrenia works through the same pathway. Except what was really interesting about this particular small molecule is that it couldn't get out of blood vessels. So when they were giving it to patients, it was getting stuck within the blood system, which was not great for trying to get something into the brain to affect neurons, but was really great for us when we were trying to get something to stay and target blood vessels. And so we tested this small molecule inhibitor in our mice. And what you can see again, here are the tumors out of the mice, and it's quantified over here, is that these tumors are markedly smaller. And if we look at the blood vessels now stained in this magenta color within the tumor on cross-section, you can see the number of blood vessels in these tumors is also dramatically reduced. But what was really exciting about this, and also really important, is we were giving these chemicals, this small molecule inhibitor, systemically to the whole body and letting it circulate everywhere. And so if we take another tissue like the liver, everything in the liver looked very normal. And so one of the things that now motivates our studies in my own lab is understanding how different vascular beds can be responding differentially to a treatment like this, which really matters when moving something from a preclinical setting to a clinical setting, especially when we're dealing with systemically, the whole system delivered small molecules. And so, like I said, now we're trying to synergize our information we know from our mouse studies with our information we learned from the fish to address some key points. For instance, can we keep animals healthy long-term with these small molecule inhibitors? You know, as Chuck mentioned, it's important that animals be able to live and grow with these things for at least a number of weeks or months, because the treatment of cancers is usually not going to be a one-shot kind of thing. And so it's important that people and animals can tolerate these molecules long-term. And then, you know, our real question is there any way we can make these more potent? And what's really the science behind why this works? And so my lab is now moving to understanding different factors that contribute to fish vessel development, predominantly how blood flow affects these, because tumors are known to have very slow, turbulent levels of blood flow. And we're wondering if that affects the expression of particular genes within the tumor microenvironment, particularly in the blood vessels. And so this movie that's playing while I've been talking is just to show you that we can image in exquisite detail. So here the blood cells are this magenta color, the blood vessels are green. So we can image with a high level of visualization what's happening to both the blood and the blood cells. And what's really interesting and what you should know about the fish that makes them an excellent model for this is that they can actually survive as embryos without blood or blood vessels and have largely normal development. So this is a zebrafish mutant shown here in these light panels, where the top embryos have normal blood vessels and the bottom embryos don't have blood vessels, nor do they have blood. And you can see these fish grow completely normally because they can exchange nutrients and waste across their skin. And so this lets us study blood flow regulated genes in really high precision without killing the animal or without worrying about cell death. And so we've now done trap-seq profiling. It's just a technique to be able to pull out RNAs that are being used by a cell at any given moment to understand genes that are dramatically downregulated or dramatically upregulated by changing blood flow rates that the endothelial cells see as they're growing. And so we've started analyzing a number of these candidates, but I'm going to talk about this single transcription factor just very briefly, just to show you the power of these types of tools, KLF2. And so what we can do is we can then take our zebrafish model and a mouse model, and we can say, where are these things expressed? And for instance, we know this one's expressed more on a vein. And so a vein of a normal animal looks very much like the vessels in the microenvironment of the tumor. So this informs, at least prior to doing tumor studies, superficially informs what we're looking for when we start moving into tumor models. And then what we can do is then look at these blood vessels in these animals that are missing, say, this transcription factor. And what we see is that there's a different association of secondary cell types with the blood vessels. So under control conditions, these green cells, which are just cells from the surrounding microenvironment, don't interact with these vessels that have these normal levels of KLF2. But if we get rid of KLF2, now we start seeing these abnormal association of cell types that shouldn't be there. So now our concept is to go into the tumor, into blood vessels and tumors, and see if we have these artificial association of cell types that shouldn't be there. So can we make a tumor vessel look more like a normal vessel to enhance the use of some of our therapeutics that we've been testing? And so that's really where my lab is, what we've been working on, what we're trying to do, and conceptually what we're working for. And so with that, I just want to say a huge thank you again to the Cancer Research Foundation, which is allowing us to interrogate this type of research. These are my lab members, our collaborators on this project. And like Chuck, having this funding has conceptually allowed us to lay the framework, do a lot of these high risk studies that we maybe wouldn't have been able to take on with traditional funding sources, and parlay this into some larger support, particularly at this point from the NIH. And so with that, I'm happy to answer any questions that come up, just like Chuck was. And thank you again for having us today.
That was terrific, both of you. Spectacular. I don't know if I'm in speaker view right now.
We've got all four of us up.
Okay, great.
So, looks like we have 2 questions in the Q&A.
Do you want to go ahead and read those,
Sure.
Matt,
So
or I'd be happy to?
Linda asks, so if you starve the fish, it loses melanoma. Won't they die in the process?
Yeah, so great question. So the time frame over which we fast them is it's not that long for them. So one of the biggest metrics for that is if you feed, if you reintroduce food they're happy and will breed again the next day, which is really that's sort of the gold standard, I would say, of a healthy animal. And so they do very well. And so, yes, it's a pretty, reasonable length of time. And very briefly, just, the fish in the wild, we think actually feeds much more sporadically than it does in a lab. Like, we feed them every day. In the wild, they probably kind of gorge, for one day here and then don't get anything for many days. So interestingly, that may be actually a healthier healthier approach in the wild. So.
And Chuck, can I ask you a slight follow-up question? Having seen you present this before, I think, would you say your goal with doing that is not necessarily to take fasting into the human population, but to identify genetic pathways that could be targeted with a therapeutic?
Absolutely. I think that's always been the challenge, is that, there's writings back into the 1400s where people realized that, some level of cutting back on the things that taste good in life were good for you, but it's hard to sustain. And so, not that, it's just a very hard thing to sustain at a level that is effective. And so we'd like to dissect. It's really there isn't sort of a central principle. I mean, there are some pathways like, mTOR and things that are of course involved, but if we could find a guiding pathway that could, maybe hit that pathway at a much higher level. And, it'd be nice if you could still have cheeseburgers too. And so.
There's another question. Is there sufficient conservation of the immune systems between zebrafish and humans to help researchers understand the potential toxicity of these therapies, i.e. off-target offsets?
Yeah,
or effects?
That's a great question. The immune systems we've found, even more conserved than we might have hoped. There was a recent study where a group looked at all the different subtypes of immune cells, and a lot of immunotherapy targets even very specific T cells called Tregs, and the zebrafish have Tregs. So while the antibodies don't always, or usually don't work off the shelf, the same pathways are involved. And so if you get rid of those cells in a fish, the fish get autoimmune disease just like a human. And if you get rid of B cells and T cells in the fish, they have an immune deficiency syndrome like a human would, or some of the mouse models where you can transplant in any old tumor and the fish will not reject it because they don't have an immune system. So it's actually pretty remarkable how similar. And I think it's just the fish is still maybe we're a little younger organism than the mouse and hasn't quite caught up there.
Yeah. And I would add, following up on that, also from the standpoint of blood vessels, the cardiovascular system between the fish and the human may be more similar even than the cardiovascular system between the mouse and the human.
Wow.
So the fish heart rate is actually similar to that of a human, whereas the mouse heart rate is about 3 to 6 times faster than a human. And so one of the things I think we've struggled with coming from mouse to human studies is that the metabolism of drugs may be faster in mice. So we don't actually see the full toxicity of drugs because of how fast things are metabolized. And so in some respects, using the fish to do that what kind of work is giving us a better understanding of breakdown of metabolites and what might translate to humans in a reasonable way.
We've got another question for Dr. Stratman. Might your work have applications for people with diabetes? We know that the anti-VEGF approach is important for reducing complications such as retinopathy, but it only works in about 50% of patients.
Yeah, I mean, I think that's a fantastic question, and something that we're really motivated by how generalizable are many of these pathways we're studying. There are a lot of earmarked similarities between what a bad blood vessel looks like in cancer, what a bad blood vessel looks like in diabetes, what it looks like in heart disease. And so I think what's really interesting and what our work is kind of dovetailing back on is that very early pathways in development that drive the formation of all of these things, like in particular in our instance, blood vessels, If we understand those pathways, then when we start looking in disease, it seems like we're almost studying the reverse of what development is. And so we've been able to use these models with a fairly high level of prediction to understand what we would start looking for in these diseases. And so I think that's a great question. We haven't looked at diabetes directly to know, but I think it's a very, very fantastic thought and something I should probably consider, yeah.
Matt, can you let Neil ask his question?
Yep, we've got probably time for one or two more questions, and we've got somebody who's raised their hand. So go ahead, Neil. You got to unmute yourself, Neil.
Got it. I'm raising my hand because I can't figure out the way to really make a question. So it's an age-related issue. Tell me what you think, each of you, or together, the next steps in application to actual really getting cures for melanoma, etc. And the second part of the question, which is somewhat tied to it, have you had any success with funding any of the drug companies that are in this space?
Yeah, so perhaps I will start. The first part of this study that I'm involved with, we started when I was a fellow at the NIH. And so we are sort of leveraging the full force of the NIH to help fund the translation of our small molecule that we've identified into a clinical trial. Now, understanding the basic science behind that is what's really stopping us from moving forward, because there's things we need to understand like toxicity and things like that. So that's where funding from a drug company really could help. I haven't fully tapped that space to try to see what kind of money could come from, let's say, the company that makes the drug we're interested in. But I think as we move forward into really considering a clinical trial, when we have it pinned down exactly how this is working and how we would move this into a human, I think that's a really great idea to loop in these pharmaceutical companies that make these inhibitors. Yeah.
And I would say as well that one of the additional areas in which I think melanoma could use some agents. So the immunotherapy is a key aspect, and then what we call targeted therapies or kinase inhibitor therapies for BRAF mutations. Those are a central piece, and I think the idea of sort of epigenetic therapies are coming into play, and I think that's where like our work will come in. And I'm involved in an NCI supported trial through the CTEP. Where there's an epigenetic therapeutic that it's not from our stuff, but the general concept of including an epigenetic regulator inhibitor with these other agents, I think will hit another leg in the be another weakness in the melanoma that I think we'll be able to hit. And coming from the idea of antibiotic therapies where you target 4 different pathways if you can, and it makes all the drugs more tolerable and more effective. And I think that's where we'll go, hopefully.
Okay, we've got one other question from Linda. So how do you translate a therapeutic to humans? That is, what's enough, what's too much, and could it be different based on gender and height, weight?
Yeah, do you want to start, Chuck, or do you want me to?
Go ahead. Go ahead.
Yeah. So, we're just in the beginning space of considering doing this, but I think there's ample, one of the things NIH asks for when you put together a paper or put together a grant for them is to consider both sexes, male and female, because there are many drugs that fail. For instance, many of the early breast cancer drugs that were screened and effective in mice were tested on male mice. And so then when they were put into the female population, they weren't quite as effective because of different types of hormones, et cetera. And it was just kind of conceptually not thought of as important as it maybe now in retrospect, like looking back, it really was. And so I think your point is extremely valid, and NIH puts a big premium on that before they release a clinical trial, release a drug to be tested in humans. Have you shown efficacy in both genders? If not, why? Have you considered how much dosing you can give to a mouse at a certain weight? And then you do a small pilot study in humans. You know, one of the beauty of working with places that work on rare disease or have clinical trials already in place like WashU is that you have, it's not great for the patient population in that they may have a rare disease or a cancer that's not responding to conventional therapies, but there's a lot of researchers like us who are trying new things. And so it gives patients an opportunity to participate in clinical science, try something, you know, to see if their conditions respond. And it really, you know, I think it's a win-win in a lot of ways for everybody, and that it gives back hope to patients who have maybe tapped out some more conventional routes. And so that's really how the earliest patients, you know, from my understanding, get involved in many of these clinical trials. And then it expands from there as you get more data to support or refute your hypothesis. So.
I think you covered it. Absolutely.
Okay, we've got one final question.
So I wanted to ask both you, Chuck and Amber, what the current landscape is for funding for your work right now, and do you see it, contracting or expanding? And then in this most recent phase, in the pandemic, have you had a sense of what that's doing to funding for science in your own labs?
Yeah, I think there's two parts to it. I think there's been a challenge of restrictions on doing the actual work that is lifted, but I think there's going to be an echo of that from earlier in the year, that a lot of, and I know we've had a lot, we've been able to open, and this is not always the case everywhere, and there's different levels of activity allowed. I think there's just a lot of there's concern, right, at the federal level that I guess the NIH budget, I guess it was even increased, but there are always these concerns that there are a risk of it contracting, and that is concerning. And just to slightly dovetail that question, I think the concern about getting folks to do the work, the best folks in the world, is becoming is a concern as well, with how visas and things are changing, and a lot of folks, their PhDs worldwide, and I've throughout my training have benefited from working with those folks. And it used to be pretty straightforward. It's getting harder, and that, I think, is going to become a barrier just as much, if not maybe even more than the dollars, I think, to completing the projects.
Yeah, I would echo that very, very significantly. That, we have phenomenal training of U.S. scientists and U.S. MD PhDs, but there's a lot of labs that need a lot of people, and being able to tap into the world's resources and recruit them to the United States is a huge advantage for U.S. science, and losing that, decreases the talent pool that we have available to our labs. The only other thing I would add to what Chuck was saying is, institutes like the National Cancer Institute with NIH are traditionally hard institutes, particularly for young investigators to get money from, because the pay lines are very low. So it just, I guess I want to use that as a chance to say again that that's why foundations like the Cancer Research Foundation are invaluable to our careers as young investigators, because it gives us a buoy and the money to do our work that we need to do, to be able to compete side by side with labs that have millions of dollars of funding a year to do their work, and get the data we need to really reach out to large funders like NIH to get the money into our labs. And so I don't know if that's going to get better or worse given the current COVID-19 situation, but I don't foresee it getting necessarily easier. And so So yeah, again, you know, opportunities to be involved in a foundation like this are really invaluable for us as young investigators. So
That's a great way to end. Thank you. Thank you, Chuck. Thank you, Amber, for not only your time tonight, but all of the important work that you are doing. And I know, I hope I speak for Cancer Research Foundation, that we are extremely proud of all you are doing, and look forward to hearing about it in the future again. Thank you all for participating, those of you who joined us tonight, and we have another one coming up on December 10th, so we will share that date with you with Lucy Godley and Michael Drazer from the University of Chicago. So thank you, and have a wonderful evening.
Thank you so much.
yeah, thank you. Thanks.
Bye.
Bye-bye.